to introduce Laurence Hurst, who's director of the Milner Centre for Evolutionary Biology in Bath, and is doing our latest Mendel at 200 seminar from evolution at synonymous sites to improve gene therapy and diagnostics. Laurence.
Lovely, thank you very much. It's a real pleasure to be here and an honour to be part of your Mendel 200 series. Excellent. So this talk is a little bit unusual because I'm an evolutionary biologist. And my main interests, I guess, are trying to understand genome organization put on a big evolutionary scale. So let's say if we're looking at the human genome, what are you actually looking at? So there's one conception which you might think of either as a sort of the hyper-adaptationist model, or the ENCODE model, if you the more genomic view, and that is when we're looking at a genome, we're looking at the most perfect Swiss watch. And the diagnostic of this is if you pull bits apart and you don't understand what a bit of it is doing, what the cog is doing, it's not that it doesn't have a function, it's because you don't understand its function. So that's one model. The alternative is not completely random crap. It works. So you're here living, breathing, doing something right. But when we're looking at a genome, is it actually more or less a heap of crap that gets the job done? Now, that's what a lot of evolutionary biologists might tell you, and it fits with the alternative to the adaptationist theory, which would be the nearly neutral model. Now, I don't particularly want to be arbitrating. A lot of what I do is try and arbitrate between those two models, but today, as I'm talking to people with a medical bent, I'm going to go from that sort of question, which is the broad underlying question, what in the genome is functional and why is it functional? But the reason I want to know this in part is, well, part of it is because it's a great debate within evolution. Is most evolution actually a neutral drift, or is it mostly selection? But in an alternative context, it's important to know because, so first off, when we go in, let's suppose you've got a kid with a disease, you sequence mom and dad, you sequence the kid, you'll find some genetic alterations, and you want to know which ones are likely candidates as being causative of the disease. Now, to do that, the first thing you do is filter and go, well, that can't be it. That's almost certainly irrelevant. You'll get about 100 new mutations. Lots of them will simply throw out, and lots of them will then hone in on. But every time you throw one out, you're making an implicit assumption about what is and what is not functional. So it's important for diagnostics that we know what is functional, what is not functional, because in that context, functional means if you change it, it's going to have a consequence that could cause disease. The other reason is that actually if we want to go for therapeutics and make better genes for gene therapy, for example, make better transgenes, then we better understand exactly what's going on. And it's partly because of this. So within gene therapy and when we're making transgenes, we can't change the protein, but we can change the synonymous sites. Now, at first sight, that's going to be irrelevant for what I'm going to show you today. It is anything but irrelevant. So what a lot of my work does is examine both gene expression evolution and synonymous sites and how they evolve to prove both diagnostics and gene therapy. Now, I'm not going to talk much about the expression, but I'll give you a very quick overview of it. One of the main problems with gene therapy is if you take a gene, put it in a vector, insert it into the genome, then it is not uncommonly the case, particularly if you put a strong promoter in front of it, that it will affect the neighbours, and the neighbours will affect it. So genes are not autonomous in their expression, but then are there places in the genome that are safer than others to insert genes into? And if there are, what would they look like? And one of the first things that we discovered a few years ago is that it turns out that the best predictor of the amount of evolutionary change in gene expression between us and the human chimp common ancestor is actually the amount of change in the gene expression of your neighbours. And this is what this particular plot shows, for example. So a positive Z here means a particular gene has increased its expression from the human chimp common ancestor to humans. A negative one means it's gone down. And then the Y-axis, you will see quite strikingly here, is the change of the neighbours. So our genes are changing in a coordinated fashion. And so some of our work is saying, well, if we know that, can we find blocks in the genome that don't show this sort of trend, where one can change, but the others don't change? And if you can, well, there's a safe harbor for gene expression. So genes most assuredly are not autonomous. And as we said, we know this lack of autonomy is important. The first gene therapy trials actually had to be stopped, and they were stopped because the gene had inserted next to an oncogene, it had upregulated the oncogene, and it killed the patients, which is of course not what you want to do. So we're using the way gene expression changes over time to attempt to define genomic domains that are safe. That's say high and low autonomy. We're looking at those with high autonomy, so I can change my expression, but the neighbours don't change, and vice versa. From that, we can define safe harbours, and from that, we can define which vectors to use. And I'll give you a quick answer. We already know that the ones that are currently being used, we probably shouldn't be using. We should be using Sleeping Beauty instead. Anyway, that's the bit I'm not going to talk about. So what I want to talk about in this talk is instead what's going on at synonymous sites. And I'm interested in synonymous sites for a number of reasons. One is for improving diagnostics, as I said. So it's classically been assumed that synonymous mutations don't cause disease. I'll show you that's absolute nonsense. They very commonly do. So we want to understand which ones do, how they cause disease, and then in turn improving transgenes for gene therapy, because we can tweak the synonymous sites if we want to. So can we tweak the synonymous sites in an intelligent fashion to enable robust gene expression? Now, just in case, I wasn't quite sure what the audience was for this, but just in case, let me remind you, because this talk would be utterly tedious if you didn't know what a synonymous mutation was. So I am now going to patronize almost certainly everybody listening by telling you what a synonymous mutation is. So of course the genetic code, 64 codons, 61 of them are coding, 3 stop codons, but only 20 amino acids. 18 of those amino acids have more than one codon, and it's mutations between those that we're interested in. So here you see, for example, GGC to GGT, that's a synonymous mutation because they both, GG anything gives you glycine. So back in the 1970s, these were called silent mutations. Silent because people presumed they don't change the protein, therefore they have no effects whatsoever. And for regulatory purposes, these are particularly interesting because we're allowed to change synonymous sites. The American regulatory authorities, for example, if you make a transgene, they're only interested in the protein content. They have to license the protein. They actually don't care about the underlying construct itself. So you can change the synonymous sites. So those are the two, what I'm going to be focusing on today. Going back to that evolutionary point of view, synonymous sites are interesting because it's part of the system that's always assumed to be part of the Mickey Mouse watch. That silent component means it's not functional. So we've always assumed that these are mutations that are not functional. So in a sense, I'm pushing that particular problem to go, well, actually, are they? Is this a part of the functional component of the genome that we never noticed? And of course, that brings in the most obvious question, if these are actually truly synonymous. Now, synonymous, yes, they are truly synonymous, but are they truly silent? Why would changing them make any sort of difference whatsoever? So what I want to do in the start of this talk is take you through some of our old work, which is now the paradigmatic example to illustrate how, in fact, synonymous mutations... can we get rid of that?
Yeah, but if you click on it.
There. Hey, gone. So I'll show you the paradigmatic example, and the amazing thing I think the example I'm about to show you is you go, it's so obvious that synonymous mutations can be under selection. You'll wonder why anybody ever thought that they could actually be genuinely silent. And the example we're going to look at, it concerns splicing, and what I'll show you is that actually when it comes to humans, we have got a very particular problem, and we've got a problem because we've got lots of and very big introns, which gives us a big problem, and we've outsourced the solution to that problem into the exons. So this is a classical textbook sort of illustration of what goes on in splicing. They always show the exons in one color, the introns in another. And of course, it's actually a computation, if you think about it, for the cell, an incredibly difficult problem. How do you know where those splice sites actually are? Because in the cell, it isn't a big gray bar and a thin black bar, it's just A, U, C, and G. So how could it possibly know the answer to that? So how can a cell possibly know where those junctions actually are? Now, if you go back to early textbooks, you'll see pictures like this. And the important things about pictures like this is you will notice that nearly all the interactions, so this is an illustration of how splicing actually happens, nearly all of the interactions, so here, for example, the U2 SNP is within the intron, and likewise the U1 SNP interacts with the intron, and there's very little going on in the exons. And this is fine, because that's actually how yeast does it, more or less. The exons are more or less irrelevant to the splicing process, and the splicing process is dictated by things binding to introns. That, however, is absolutely not how we do it. Our problem is much deeper, and here this is going to be the stars of this particular story. So here again, your big black one, the big ones, they are the exons. The bit in between again are the introns. And the stars of this story are these things here: exonic splice enhancers. And exonic splice enhancers are so defined, so called because they're exonic and they enhance splicing. It sort of says what it is on the tin, as it were. But the important thing about these ESEs, there's many particular points about, they are parts of exons. They are motifs that sit in exons. But that means immediately that your exons have got two jobs to do. They've got to specify the amino acids for the protein, and they've got to specify also I'm an ESE, which means they've got two jobs to do. We call it dual coding. What ESEs actually do is they bind these serine arginine-rich proteins. R is arginine, which I always think is, it's like, you know, pirates named amino acid, arginine. Anyway, serine arginine-rich proteins, SR proteins. SR proteins bind to ESEs, and the binding of an SR protein to an ESE then recruits factors which say there's a splice junction somewhere near here. That's more or less all that's going on there. But they're odd. So these SR proteins are typically these ESEs are typically just 6 nucleotides long, and you might think, well, how can something 6 nucleotide long be interesting? But they have a number of very intriguing properties. First is they're greatly enriched towards the ends of exons. As you'd imagine, these are things that are being sticky for SR proteins. The SR proteins stick to them, and that says there's an end of an exon somewhere near here. So they're enriched at the ends of exons. But they're also dose sensitive. So the more ESEs you have at the ends of an exon, the more likely it is that an SR protein will find it, and the more likely it is that that exon is going to get spliced accurately. So they're enriched at exon ends, and you go, well, what sort of dosages are we talking about? Well, at exon ends, the average dose of ESE is about, we estimate, between 30 and 40%. So that's to say, if you take one base pair somewhere towards the end of an exon, and that's up to about 70 base pairs for an exon, 30 to 40% of the time, that will be part of an ESE contributing to SR protein binding. So these are big plugins in our genome. They're incredibly common. They also have a very strange nucleoside content. 50% of the nucleotides in experimentally demonstrated ESEs are A. So the null would be about 25%, but 50% are A, 25% are G. So more or less, these are purine-loaded motifs. And as I said, they're enriched towards exon ends, both 5' and 3'. And one of the original observations was Chris Berger's group in 2004 that got us really intrigued is they observed that the SNP density is lower as you move towards the ends of our exons. This is something we've been able to replicate as well. And it's because there is purifying selection operating on mutations in ESEs. So as it happens, there's an interesting corollary of that, which is that actually humans are most similar to each other at the ends of our exons. But this is a way then to think about human exons. Human exons towards their ends are fly paper. They're fly paper for SR proteins. So they're sticky to attract these SR proteins to say, look, here's the end of the exon. This must be an intron because we've accumulated these things here. So it's as they are towards the ends of our exons, we're flypaper for SR proteins. But one of the more important things I think to notice about these is we know they function up to about 70 nucleotides away from an exon end. 30 to 40% of the sequence at exon ends of this 70 base pair domain are ESCs. But the average exon size in humans is actually less than 140 base pairs, which means that any exon that is less than 140 base pairs is actually all exon ends as far as an ESE is concerned. So a strikingly high proportion of our genome is in these domains that are flypaper for SR proteins dictated by ESEs. So our first thought was, okay, so the textbooks say there is no selection on synonymous sites in humans. I can tell you some of the history of that, but that's always been the understanding. And we thought, well, okay, surely there must be. If you've got these motifs, 40% towards the exon ends, they don't care what frame they're in, they can be in any frame. Surely if they're there and they're functional, that means that whenever they're hitting a synonymous site, 2 of every 6 are going to be synonymous sites on the average, something like that. Well, there's going to be purifying selections there. You'll have broken an ESE. And so we thought, okay, well, why don't we just simply go comparing as we go towards the end of an exon? So that's why we've gone up to 70 base pairs here. So this is large numbers. I think in this case, it's mouse rat exons. And for each orthologous pair, then we can say, okay, do you have a synonymous difference between you? How far away are you from the border? And we just look at across all of these. And what you see is that if we now split any given change or any given bit of sequence as you are ESE or you're not predicted to be ESE, then it's not even a subtle effect. The ESEs evolve about 1/3 slower than the non-ESE does. So this is the point where we go, oh, look, we have really discovered something quite important here. Remember, synonymous sites are not under selection in humans. This is absolutely, they are. And it's not even a subtle effect. You can very easily detect it. And so we estimate that overall, when you factor in what proportion of exon is towards exon ends, what the extent of the reduction is, et cetera, it causes about a 20% reduction in the rate of synonymous evolution. So if you try and estimate the mutation rate between mouse and rat, for example, assuming that synonymous mutations are simply neutrally evolving, you'll underestimate it by about 20%. So it's good. It's not a subtle effect. You can very easily detect it. When we actually submitted this one off to the top molecular evolution journal in the field, we got back referees reports said, this is very interesting, quite intriguing. And the editor said, I don't care. It is well known that there is no selection on synonymous mutations in mammals, and he rejected the paper. We argued the toss and got it published anyway. But that's how hard the prejudice was built in, that there cannot be selection on synonymous sites. I think the logic is very clear. And you can then go on and it predicts all sorts of things. So it predicts, for example, that if you've got really short exons, then nearly all of the exon is going to be under this constraint, whereas if you've got a longer exon, it isn't going to be, because the middle can fairly easily evolve. And that's exactly what you see here, that shorter exons, so higher proportion within 70 base pairs of an exon end do indeed evolve slower. The other thing that's very nice is we can explain codon usage. So as you move towards the ends of our exons, what we're going to expect is the codons are going to change, that they're going to be selected to be stickier. And so here you see, for example, one pair, GAA and GAG. This is at the 2 ends of the exons, 5' and 3'. And as you can see, so despite these things being synonymous, GAA is preferred over GAG as you move towards the end of an exon. Now, that's exactly what we would have predicted, because GAA is, in fact, the stickiest of the sticky. So if you have the sequence GAA, GAA, it is as sticky can be to SR proteins. GAG is pretty sticky as well, but GAA, GAA is as sticky as they can. So what we could show was that we take the 59 codons that have got at least one synonym, 47 of them will show some trend like this, and 42 even after the great destroyer that is von Frani correction. So most codons show a trend either being preferred or being avoided as you move towards exon ends. Then the question is this anything to do with ESCs? So we have a rather pompous Z-score. It's just a Z-score, but we call it the hexamer preference index. And that basically measures how sticky you are, how much you liked in ESEs compared to null codon usage. So a high score means you're greatly enriched in ESEs. And what we can then do is compare each pair of codons. So each letter here is a comparison between a pair of codons. And the prediction are twofold. First is that the extent to which you get a difference in preference of usage, which is what you're seeing on the y-axis, that should be higher if there's a bigger difference between the two codons and their stickiness. And that's what you see. So there's overall a negative slope. And we also expect that the one that is stickier should be the one that is preferred at the exon end. So they should sit below the line 0. And indeed, they typically do sit below the line 0. There's a very interesting analysis to be done about the very few that do sit above the line. They look as though they're avoiding cryptic splice sites. So there's a second selection going on there. So that's what's going on at synonymous sites. Then, of course, there's the question, does this affect proteins? Does it mean that our proteins are also actually not the best proteins? Could it be that our proteins are actually having to do two jobs as well? So we've seen our synonymous sites, they've only really got one job, which is be ESE or not don't be ESE. But what about the proteins? Are we affecting proteins as well? So we did a very similar analysis. This is now looking not at rates of evolution at synonymous sites, but looking at amino acid changing mutations. And you see exactly what we'd have expected as you go towards the end of exons. So here we are going towards the end of exons, that the rate of protein evolution goes down. Yeah, exactly what you'd expect if they're being under selection to be preserved for ESEs. I'll show you more evidence that's what's going on. There's this funny little statistical effect that the ones down here could be short exons, the one up here long exons. So this controls for that, and you still see it. The most interesting are these though. So there's a bunch which go the other way. And what these are retrogenes derived from intron-bearing parental genes. So these don't have any introns. What you have here is the distance from the exon boundary in this case is the distance from the exon boundary that existed in the parental gene. So our retrogenes actually have an evolutionary memory of where the boundary used to be. And actually what's going on here is they're being released from that second constraint of having to specify where the ESE should be and where the SR proteins need to bind, because they don't have introns. And so they're trying to get rid of at least some of them. And this is one of the observations that led us to think, now that could be interesting, because when you're making a transgene, you typically get rid of the introns. Our transgenes mimic retrogenes. So does that mean we've got a clue as to how to engineer a better retrogene, a better transgene, because we've taken away the introns? Do we now take away the sequence which says there's an intron there here? I'll come back to that at the end. Again, a bunch of results which I think are very straightforward. Is this slow evolution towards exon ends anything to do with ESEs? Yes, it is. So if we partition them ESE versus non-ESE, the protein changing mutations that also change in ESE happen less commonly. It's not mutational, it is simply selection. And it turns out this is also then a very good predictor of the rate of evolution of an exon. So if you have an exon that's a short exon, all of it's next to the boundary, then that exon is a very slow evolving exon. Whereas conversely here, we find fast evolving exons, and our retrogenes, of course, are relatively fast evolving. They move, losing these constraints. Again, there's a problem. These exons are from different genes, but if we look within an exon, which is what this figure here is showing, and we can compare the center of an exon's rate of protein evolution with the flank of an exon's rate of protein evolution for long enough ones, we find that the center is evolving 30 to 50% faster than the flanks are. Again, it's not even a subtle effect. So to reiterate, we are most similar to each other at the ends of our exons, both at synonymous sites and at protein changing sites. The other thing I want to emphasize about this very much is that this is not a small effect. I mean, I sort of emphasize we could easily detect it at synonymous sites, but we only actually have one universal predictor of the rate of protein evolution, and that is how much is the gene expressed. Highly expressed genes evolve slowly, lowly expressed genes evolve a lot. So if we take the rate of protein evolution here, that's X, Ka, that's our measure of protein evolution. Y can be what proportion of a given gene sequences towards the end. Z is our breadth of expression. And then we can say, well, how well does that tidiness of the gene, how small are the exons effectively, Y, how well does that predict protein rates of evolution compared to our other best predictor? And this is a universal best predictor. Bacteria through to every other form of life, breadth of expression or rate of expression is the best predictor. So we get a correlation between protein evolution and breadth of expression of about -2.1. And for the exon structure being small, it's about 0.2. We can do the partial, so we can control one for the other, and we do that there, and they come out about the same numbers. And that's because, and this is quite an interesting observation that no one, I think, has understood, and I still don't quite understand it myself, that our broadly expressed genes, our housekeeping genes, have small exons. Who knew? Very strange observation, but the more important thing about, not only do we not understand quite why that is, the more important observation here is that this simple exon structure effect, small compact exons are slow evolving exons because they also have to be specifying their splice instructions, is as important a predictor as our best predictor of protein rates of evolution. So it's not even a subtle effect. Does it then skew the amino acids that we use? Well, I do apologize for complete data overload on this. What you're seeing here, and what you should be impressed by, is the number of the numbers here that are in yellow. So what you're looking at here is for each amino acid, 5' and 3', so there's the two columns, 5' and 3', we also separate out the four-fold block of leucine, serine, and arginine, and the two-fold block of leucine, serine, and arginine. So the three at the bottom there, L, S, and R, those are the two-fold blocks of leucine, serine, and arginine. If after bond for any correction, an amino acid has a trend in its usage one way or the other towards exon ends, then it is in yellow. That's after bond for any correction. So this is 46 comparisons. The take-home message of this is they're nearly all significant. So nearly all amino acids are showing some sort of trend. And you can say, well, could that be some structural motifs that are avoided or something like that? To which the answer is no. We know the answer, well, at least not in its entirety. But we know that it's something to do with the nucleotides because we split, for example, arginine. So here you have the 5' behaves exactly the same way. Here you have the four-fold degenerate block of arginine usage on the left panel and the two-fold block of arginine on the right panel. So they're both specifying the same amino acid, but one is being preferred towards exon ends and one is being avoided at exon ends. So it can't be anything to do with being arginine. It has to be something to do with the underlying nucleotides. So, okay, well, our prediction is very obvious that the one that's being preferred is the underlying codons for it are stickier, and the one that's being avoided, the underlying codons are not sticky enough. So we can do exactly what we did before, and it turns out that's exactly what you find. So what you see on the y-axis here is if it's a positive row, that means it's being avoided as you go towards an exon end. Negative row means it's sloping upwards, so it's being preferred at exon ends. And as you can see, generally speaking, we've got a very nice ability to predict which ones and in which directions are significant. It's down to the underlying stickiness, which is what the y-axis is telling you. It's down to the underlying stickiness of the underlying codons. How well do they stick SR proteins? And rather nicely, there are two arginines. So the four-fold block of arginine sits at one end of the distribution, really not liked. It's really not sticky at all. The two-fold block is one of the stickiest blocks, highly preferred at exon end. So we think most of these amino acid trends, we don't have to be evoking any avoidance of structural motifs as you move towards exon ends or anything like that. It is simply you have to be sticky as you go towards an exon end, and if the underlying codons are not, you've got a problem. Now, the summary of all this is, I don't think I've told you anything that you didn't actually already know. You might not have known the details, but the general principle, I think you understood entirely. Because if I were to ask you, why does something like Tetra Pak have those folds underneath? I could have given you a hyper-adaptationist answer to say, well, next time an earthquake hits Bristol, because of those folds underneath, the center of mass is slightly lower, which means that when the earthquake happens, it's slightly less likely to fall over. That is true, but it's not the reason that you have folds underneath the Tetra Pak. The folds underneath the Tetra Pak are a hangover of the manufacturing process. You know how Tetra Pak is manufactured, they sort of simply fold in the ends. So all I'm telling you is that our proteins are not just simply the vessel, they're not simply the product, they're also containing the signatures of the manufacturer. And that's exactly the same principle as dual coding that we saw for the synonymous sites. These are representatives not of the end product, but of the manufacturer of the end product. And in that sense, I don't think I've told you anything new. So it's about not only what the protein does dictates its evolution. Put differently, there is no point in having the world's very best protein if you can't make it. If you can't splice it properly because your ends are no good, then there's slight you'll just say, I cannot operate on that because you can't actually make it. Now, the other thing I want to particularly reiterate, as I think I already mentioned before, is that humans actually, in this context, we are odd. We are quite the outlier. So usually we think of humans as just, within an evolutionary context, another species. But in this context, well, we're representative of large-bodied, slow-reproducing, small population-sized species, but we are, when considered en masse, rather odd. And that is because we have got particularly long introns. So mammals as a whole, and vertebrates as a whole, more or less all have conserved exon sizes. Our exons are unusually small, but and that's because we have many, many introns. So we've got a very high intron density, but our introns are even larger than, for example, mouse's introns. So we have particularly large introns. So if you ask what is the mean coding sequence length divided by the total gene length, ours is about 18% or so. So if you take a gene, only about 18% of that is coding sequence, the rest for us is intron. Now you go down to that other extreme. So remember that other splicing model I gave you, which was from Cerevisiae, they don't have this problem at all. If they've got introns, they're tiny, compact little things, and they will splice themselves out more or less. But what you see overall is that as the figure on the x-axis changes, so that the introns are getting bigger in comparison to the size of the exons, so the exons get loaded with these sticky exon ends, and that's what you're saying. So it doesn't quite matter. We've got the proportion of amino acids showing a significant trend here, but you can also say what proportion of the sequence matches known ESE, and you get a very similar trend here. So we sit at the funny end of the distribution. We've got a particular problem, and our problem is that we've got big introns. And if you again think about this computationally, this is a difficult problem for a cell. Our exons, the bits that we actually want, are the small bits, sorry, the exons are the small bits in a sea of rubbish. And we know this is a problem for many different sorts of analyses. So one simple test you can do, and this was done back in quite a bit in the 80s, is you just take an intron and you put in a load of rubbish DNA into it. And it turns out when you do this, the splicing rate goes down, and it doesn't matter what DNA you put in. Just by making it physically bigger, you're making more distractions for the system. You may well be putting in funny little splice sites you never knew about, and so on and so forth. So we know experimentally that just putting something in makes it harder. We also know, so if you look at functional genomic evidence, and you find an exon that's found in one tissue but not in other tissues at a low level or something like that, these are nearly always associated with large flanking introns. Likewise, if you go between species, so you go, well, I found this exon in humans, in a kidney sample. Is it there in chimp? Is it there in gorilla? No. Those sorts of exons, again, are flanked by large introns. So these large introns are probably giving us a problem. What all of this says is we've got a problem with large introns, and large introns mean that we degrade the splicing process. It's very hard to accurately splice an exon next to a large intron. So the solution we've come up with is this. So this is human data. So we ask, what is the density of ESEs at an exon end, given the size of intron that flanks it? And what you can see is we've got a very simple solution to the problem. If you've got a large flanking intron, you have a higher density of ESEs at the flank. And so for the relatively shorter introns, down to about 25% of the sequence, is ESE, moving up to more like 35% when you've got a much longer intron flanking you. So long introns are our bugbear, they're our problem. We solve it in part by building in these ESEs, but then of course comes the problem, well, if we keep on having this problem, what happens if you have a mutation that disrupts an ESE? Is this going to be a cause of disease? To which the answer is, we know from a number of case histories, the answer is yes, and then there are now at least 100 different genetic diseases that we know that are associated with mutations, and you can show what they do is they disrupt an ESE, they cause a missplicing event, and if you think about it, this is why we can detect synonymous mutations causing disease. They were never silent. They're actually very traumatic events for that exon. Often you completely ablate that exon from the ending gene. There's nothing particularly silent about that in terms of its effects on the protein. It's completely changed the protein because it's completely changed the splice forms. So we thought, okay, let's see if we can do this a little bit more numerically. Can we try and estimate the proportion of all disease-causing mutations that cause their disease by affecting splicing? By affecting splicing, we mean one of two things. One is that there are well-known effects just at splice sites themselves. So the splice sites are sort of 2-3 base pairs around the splice site in the exon. Been known for ages that you get splice site mutations. The exon just simply is not spliced properly and you get a genetic disease. That's nothing new. So what we're doing here is we're taking ClinVar database SNPs and we're partitioning them. So the blue is the null, the red is the observed, and we're partitioning these disease-causing SNPs by their location within exons. So the nearest bar here is the actual splice sites themselves. This then is within the ESE functioning domain, and then this then is sort of what you might think of as exon core. So I said red is the actual frequencies and blue is a null. So the blue is the null derived from giving your underlying nucleotide content, et cetera, assuming a uniform mutation rate across an exon, which from parent offspring sequencing we think it is more or less uniform. And what you get is, yeah, there's enrichment of these things at splice sites. Yeah, that's nothing particularly special. The more interesting result then is this comparison here, because what you see is that in exon cores, you have almost half the number of mutations, disease-causing mutations that you'd have expected by chance. And that's because we think exon cores have nothing to do with splicing to a first approximation. Where are the mutations that cause diseases? They're gracefully enriched in these domains towards exon Ms. And of course, these null simulations here, these null amounts allow for exon sizes and blah, 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 and blah, blah, blah. So yes, not only are SNPs rare, so we're all most similar to each other at our exon ends, when you do get a mutation that disrupts exon ends, then they are more likely to be genetic disease-causing ones. So if we make some assumptions about, let's suppose that the background rate comes from exon cores, then we'll get one rate. So the end of exon disease-causing genes is, we can then get another rate, and then we can, get a total number of base pairs. From that total number of base pairs, if the underlying rate is the core rate, then we expect that number of just shy of 5,000 disease-causing SNPs. We actually observe 6,775 disease-causing SNPs. So that gives us an approximate frequency of about a quarter. So this is assuming that the core is giving you a background rate. That is probably not true. There are some mutations, even in cores, that can affect splicing. They're not so common, but they can happen. So the 24% figure is a sort of lower bound estimate, assuming those mutations that we're seeing are enriched at exon ends because they're having an effect on splicing. You might think that's a little bit outlandish, but actually it agrees with a number of much more detailed studies. So what I'm showing you here is each of these papers provides an analysis typically of one exon, and they will mutate the exon and go in a mini-gene construct. And having mutated the exon in a mini-gene construct, they'll say, okay, what proportion of the mutations have actually changed the splicing? And so that's what you see on the x-axis, the percentage of sites that appear to be involved in splicing. and it runs everywhere from about 20% for some exons up to about a limit of about 100% for others. This sample, however, is highly biased, and it's highly biased because to make those mini gene constructs, people use short exons. And so we think they're overestimating the number, but the average, when you take about this and then you extrapolate it more broadly, you actually end up with about 20 to 30% as a very similar figure for mutations that would affect an exon and cause an effect on splicing. As it so happens, this is one thing we've been working on quite recently. It led us to the suggestion that actually nonsense mutations are not nonsense mutations. Nonsense mutations are actually really interesting if you think about them, because nonsense mutations are purine rich. PAA, TAG, TGA, yeah, they're purine rich. But in at least one frame, they cannot exist in an ESE. Because if they existed in an ESE in that particular frame, remember ESEs can be used in any frame, then that would mean that the ESE can't be an ESE because the ESE is defined as being exonic. So there must be one frame that they can't work in. What that gives you en masse is that ends of exons and ESEs are particularly likely to mutate to a stop, which cannot be a component of an ESE. So stops are going to be enriched in non-ESE motifs, but very easily generated by ESE motifs because they are purine rich. So they're purine rich, but ESEs can't have stops, but that means that a mutation in an ESE to a stop is very likely to break the ESE. And that's exactly what we find here. And we again estimate, in this case, we estimate just over 33%. So we get a great enrichment of nonsense mutations towards the ends of exons associated with disease, but much less than they exist in the core of exons. And we could show a couple of examples where we could actually show that the effect of the nonsense mutation was in fact to disrupt ESE. It causes a missplicing event, so this paper will also present a mini-gene model to do that. And then this led us to another possibility, which is, are there certain exons, is there a flavor of exon that is more likely to be an exon that if you have a mutation in it, is going to have an effect via missplicing? And so again, we went back to our CLIDVAR and looked at SNPs that are associated with disease-causing SNPs from CLIDVAR that is associated with what are the properties of those exons. And the properties of those exons is they look as though they're, quotes, what you might call fragile, and they have properties that give them a very low ESE density. So it turns out it's not actually the exons next to big introns. Those actually have a lot of ESE, so probably if you mutate one of those, there's one sticky enough next to it, which will be all right. But if you don't have many ESEs at the end of your exons, mutate one, and proportionally that seems to be having rather a large effect. So the biggest predictor of disease-causing SNP density when we look across exons is actually the ESE density. So if the ESE density is low, then you're more likely to be a mutation. A mutation in that is more likely to be a disease-causing mutation. And there's lots of other signs that these are something to do with splice disruption, aside from the fact that they're associated with ESEs. So yes, so certain exons appear to be more fragile to this than others, but it's not the ones next to the bigger introns, it's the one next to the smaller introns, and that have low ESE density. So that then takes us to what we're currently doing at the moment, and that's trying to use approaches to say, can we now predict a mutation at a synonymous site that's actually going to be disease-causing? So we've got some fingerprints here of which ones might and which ones might not. We know that they tend to be towards the ends of exons, for example. The current model is working okay, but I have a little bit of a problem with it, which is we have to train it on a data set of well-described mutations that cause disease, synonymous mutations that cause disease, rather. And the problem with that is the well-described ones, almost all have been verified because they have an effect on splicing, because splicing effects are easy to verify. So it may be an entirely circular process. So to put that again, we have a class of synonymous sites that we know are associated with disease, and most of them are doing it via splicing, because that's the thing that's easy to demonstrate. We train our data set on that, and guess what? It comes back predicting, oh, splicing is probably the major mode by which these things are happening. Well, it probably would, because that's what it was trained on, because I think probably a bias, well, maybe a bias in the data set. But it turns out we don't actually need machine learning at all. It turns out actually they're just 3 parameters and we can predict fairly well which synonymous mutations are causing disease, and they tend to be highly conserved, the gene tends to be highly expressed, and the various methods say they have an effect on splicing. And with those 3, we can explain quite a lot. But we know there are other mechanisms out there that I haven't talked about. So, for example, a synonymous mutation, they will interrupt, they will generate and destroy microRNA pairing sites, and a number of those have been shown to be important in cancers, for example. There's recent evidence that some synonymous mutations will disrupt M6A, they can disrupt RNA splicing, forcing long stem loop structures, etc., etc. But the other stuff that I wanted to finish off with is then looking at, can we use any of this information? As we got this clue that there's been a natural experiment which is taking our genes full of introns and making them intronless, which may be able to give us a clue as to how to make genes for gene therapy. The major problem with genes for gene therapy are, well, one is immunological, that's nothing to do with my end of things. But the major problem is most of our genes are big, and they're big because nearly over 80% of them is intron. So we can't handle things that big. So we get rid of the introns, but if you get rid of the introns, you've got a new problem, which is that they typically won't express. And so that's been known for ages now, that if you take away introns from genes, they typically don't express. So our question is, could we learn from how synonymous sites evolved to be able, particularly in ESEs, for example, to be able to actually rationally design and better engineer? And as I said, actually what we do is even better because we use evolution, because evolution's answered the question for us. We have actually got native intronless genes. So what do native intronless genes actually do? And here we got our first big clue. We actually know that they are conserved at ESEs, they are conserved at SR protein binding sites if those SR protein binding sites are associated with nuclear export. They are not conserved when the SR protein binding is not associated with nuclear export. But this was the big clue. So how many exons does your gene have? So one is our interesting ones. These are our intronless ones. And what you notice is they've got a very high GC content. I mean, look at that GC content. They have a GC content of about 80% or so. This is outrageously high. This is telling us something quite profound, that anything in the human genome would have a GC. This is its synonymous sites. So GC4, that's the 4-fold degenerate sites. So they've got 80% of their 4-fold degenerate sites are Gs and Cs. And it turns out this is generally true of first exons. So no matter which gene you go to, so here's our first exons, and they have this very high GC content at 4th site. But all of our other exons do exactly the same. So that first exon is very, very GC rich. So there was our big clue, that there's something going on at the first exon and the 5' end of our genes to make them GC rich. And it turns out, so we could then double check this, do our successful retrogenes actually evolve a higher GC content at their 5' ends than the gene that they came from? So they were derived. We know the gene that they were derived from that contains introns. And so we could actually compare them. And what you have here is the parental genes have one content, and almost without exception, the retrogenes evolve a higher GC content at their 5' ends. We now understand why this GC content, by the way, matters. I'll go on to that shortly. But the first trick, which we did with Greg Kudler's, then go, okay, well, here's our intronless transcript. We'll make GFP. And what we're going to do with our GFP is simply change the synonymous sites. So it's always will be GFP, we'll just change the synonymous sites. And as you can see, we can completely kill a gene, and we can kill it by giving it a high AT content. Give it a high GC3 content, and this is again just at synonymous sites, and the thing can express very well. And these effects, again, they're not subtle. This is not a minor effect on gene expression level. This is orders of magnitude difference in expression level just by changing the synonymous sites. So yes, it works. Very simple little trick to get your intronless transgenes to work. You just follow what retrogenes actually, the successful retrogenes actually did, which is evolve high GC content. What is the mechanism? molecular biologists usually want to know this. We know what the answer is. So it turns out that if you don't have a high GC content, a big component of this is you don't get out of the nucleus. And it turns out that for intronless genes, there are actually 2 export channels. Our nuclei have 2 export channels, and it is effectively exactly like passport control. So if you are fortunate and Danish and you fly into Copenhagen and you show your EU passport, you just wave straight through. Those are our big genes with multiple exons and a GC-rich 5' end. You're straight through, no questions asked. If you are AT-rich and intronless, you're basically saying, you don't look native. You get strip search, and that's the other export channel, and most of the time you don't get through the other export channel. So we've got export channels to go, yeah, you look like one of my transcripts. And GC richness we are using simply as a signal, you're one of my transcripts. And AT richness says, no, you're probably just a random load of spurious rubbish that's come off the genome. I'm not letting you out of here. So we've got 2 specialist export channels which are trying to work out, are you native or are you not native? And we're using GC richness to affect that. There is, it turns out, a number of other effects. There's actually a transcriptional effect. So there's the RNA polymerase II will actually fall off. It won't process down an AT-rich 5' end as well. And for longer intronless genes, we have a specialist anti-retrotransposon system called HUSH, and HUSH goes and sits on you and basically shuts you down. So there is still a problem with very big intronless things. Big intronless things that are A-rich, you cannot get to express. It looks for, are you big and are you A-rich? If you are, you're going nowhere. So that was that paper. And then, of course, this gives us just a very simple mechanism to go and say, okay, well, we can design a transgene for you now. Give us your gene and we'll work out with a couple of tweaks probably what the best design of transgenes are. Now, if any of you have ever made a transgene, you'll know what you usually do is you go and give it to the synthesis company. They do something rather mysterious called optimization, which is as about a black art and voodoo as you can imagine. There is no sense in which they're actually optimizing your genes whatsoever. Nobody can actually work out what they're doing. If you look at the constructs that you get back from different quotes, optimization programs used by different companies, they're not similar at all. The only thing they have in common is that they avoid very rarely used human codons. That's about it. That's the only thing. And it is not even clear that when they quote optimize, the expression level even goes up. Quite a lot of the time it doesn't. We think that's actually what's going on most of that optimization. If it works, it's actually because it's mimicking these filters that we've got for good and bad ones. We actually tested that out by going, we're going to compete our version of a transgene against quite commercially optimized ones, and ours, bingo for us, ours outperformed the commercial ones quite considerably. So the summary of all of this is that evolutionary biologists can be useful. And understanding the causes of selection on synonymous sites has allowed us first to predict which synonymous mutations will cause genetic disease. And I think where we're going next is, can we apply that to parent offspring trees? You have 100 mutations. Can we say, actually, that synonymous one there, you should go after that one. You might have ignored it otherwise, but actually we think it's important. And we can better design transgenes for gene therapy. And where we're moving to now is it turns out that bacteria have completely other sets of rules. So we're trying to work out what those rules are. They don't have nuclear export because they don't have nuclei. They've got completely other sets of rules for how to get a transgene to work. And we've got some transgenes now which work really, really well in bacteria. But interestingly, they're AT-rich at the 5' ends. And that just leaves me to say thank you. These are just some of the graduate students and postdocs who did all the work. A big thank you also to Greg Kudler, who I collaborate with on the experimental side of things. But I've just been blessed through many years of having stupidly talented graduate students and postdocs who do all the hard work. And I just think great thoughts. Also, I'd like to believe. Anyway, thank you ever so much for your time as well. Happy to take any questions.
Looks great. Questions, Lindsay? I was just anxiously thinking, how do I get the questions online? But Lindsay's there.
just before 5 o'clock.
I got to show how hopeless I am with the technology.
Thanks very much. I've just got maybe a naive question. I mean, so if I remember rightly, right, sometimes you get cases of alternative splicing that are adaptive, not defective.
Yeah, sometimes, yeah.
Deliberate way of
Absolutely.
different proteins from a single gene.
Yeah, absolutely.
How do they factor into this story?
Interestingly, they don't. Well, they do and they don't. So one question is, are ESCs at all controlling that alternative splicing? The answer appears to be no. What the conserved alternative splice events have in common, so if you have an exon and it's a conserved alternative splice, then it is a very, very slow evolving exon. And the synonymous, so we have a number of exons that are conserved alternative, and they do not evolve through the primates at the synonymous site, nothing. So there's clearly some information in there, but the rate of evolution of ESCs, when we consider them overall, compared to the other sites, it's also about a third lower. So they don't seem to be that crucial in these deterministic switches. Now that is so-called conserved alternative splice forms. These are the ones that are evolutionarily important. So for example, there are decisions made when a cell becomes a neuron as to which sort of neuron it becomes. And there's a couple of genes that it has particular conserved alternative splice forms. And if it flips to another splice form, it will become one sort of neuron. If it flips to the other splice form, it will become a different sort of neuron. And those clearly are functional. If we look at the great body of alternative splice forms, however, those tend to, not to be at all conserved. So you say, okay, can I find this alternative transcript in close relatives? Usually not. And what we find is that the species that have got a lot of rare strange splice forms are species, those rare strange splice forms typically are frame shifting. They typically do not have conserved splice sites, and they are more abundant in species with low effective population sizes. So all of this says that actually probably most of those splice forms that you see, when they're not conserved, are genomic rubbish. We actually think this is one of the other great sources of transcriptional rubbish, that we have all sorts of techniques using synonymous sites to say, you're not mine, are you? So I've got NMD as a very obvious one, but there's layers upon layers of filters. So most alternative splice forms we think are just rubbish. The ones that are conserved are very interesting. They're conserved throughout the exon. And in fact, if you do an analysis, you know about Ka-KNS analysis, I'm sure. Okay, so Ka-KNS analysis is the classic way we look for positive selection operating on proteins. So what it says, Ka is the rate of protein evolution, controlling for this, that, and the other. Ks is the rate of synonymous rate of evolution. And if your metrics are right, if you've done your calculations right, if it's perfectly neutrally evolving, a mutation that affects a non-synonymous site, so a protein changing one, should be as likely to go to fixation as one that changes a synonymous site. Yeah, that's effectively the definition of neutrality. So the Ka-Ks ratio should be 1. Most of our genes have a Ka-Ks ratio about 0.1, i.e. 90% of the protein changing mutations are kicked out because they're deleterious. And then positive selection is indicated by that ratio greater than 1. So if you look at a number of our immune system genes, for example, then faster evolving at the protein level than you would have expected given their background rate. Now, if you do a scan across large numbers of genes looking for windows where Ka by Ks goes above 1, more of those are associated with the dip in the synonymous rate than actually an acceleration in the protein rate. And they are associated with the conserved alternatively spliced exons. So something in synonymous sites appears to be operating to say, yes, you're an alternatively spliced exon, and this is how you're going to do it, but it doesn't seem to be mediated by ESCs. It's been mediated in part by the synonymous sites, but nothing to do with ESCs as far as we know.
Thanks. That was fascinating. Thank you. In coming here, sorry, I should have said I worked as a clinical geneticist. In coming here, I was trying to think of what sort of mutations might be important as synonymous, and as well as miRNA. I was thinking one other which I'll ask, but had no idea about what you've been talking. I mean, before you were talking, it is fascinating to know that mechanism. The one I wondered about, is there any difference in the frequency or availability of transfer RNAs that affects the speed at which the protein is produced, and therefore could that translate into
I'm glad you asked that, because historically this is where the whole field has been. So if you go to bacteria or fruit flies or whatever, and they talk about codon usage bias and selection on synonymous sites, it's all about the codons adapting to the tRNA pool, either to make the ribosome move faster or to make the translation process more accurate. Because if the ribosome has to stall, it could be the wrong tRNA that gets incorporated. So whenever people have talked about selection on synonymous sites, and this is why the editor says we know that this doesn't happen, that selection is a very weak selection, even in bacteria and what have you. All you're doing is minorly changing the speed of a ribosome. So when we look for those symbols, the classical fingerprints of that sort of thing in humans, and that is the highly expressed genes have stronger codon usage bias, and that codon usage bias is predicted by the tRNA pools. We don't see it in humans at all. And that's also what we'd have expected, because we have low affected population sizes, so selection is a weak force. And so that form of selection, which is very, very weak selection, you're just modifying the speed of a ribosome, you're not changing the complete protein structure. That is very weak selection. So that's why people said, well, you won't get any selection on synonymous sites. But what they were doing was assuming that the selection must be mediated by tRNA availability. Now, there is then a second question. So we know that overall, we don't see that effect at all in humans. But what is quite possible is there may be an effect of co-translational folding. This does seem to happen, whereby we do have some evidence of series of codons, which probably, but we're not quite sure, the RNA ribose-seq data suggests it might be going on, slows the ribosome. And the effect of that is to allow a different fold on the peptide structures as it is coming out of the exit tunnel. Yeah, so the protein has already been formed coming out of the exit tunnel. The ribosome now pauses, and because it's just paused, the thing that is being folded now has more time to fold. And so there's some elements of so-called cross-translational folding that mutations then affect the speed of the ribosome, can then affect the protein fold. But overall, we don't see a strong connection between codon usage in our genes and tRNA availabilities, but there may be micro effects like that.
I'll stop Tom going to ask his question.
Yeah, so I was interested in your predictions of the disease-causing effect of synonymous variants, and so I was wondering whether you'd use the stickiness and distance from exon boundary as predictive features, and if so, how predictive they were compared to, for example.
Yes and yes. Yes, they are part of that overall prediction that we have. There is, however, another difficulty, which is that because the data set is very heavily skewed towards mutations that cause disease via splicing, because that's what we can identify, those, as we know, are closer to exon boundaries anyway. Yeah. And so the model ends up predicting what it was trained on. So that's why I'm somewhat skeptical about the machine learning approach, because it requires us to, we have to say, and how far are you from an exon intron junction and so on and so forth as one of the input variables. But yeah, that comes out as a variable. It's actually not one of the bigger variables, but that again may be for completely other reasons, and it's that actually genes with very large exons tend not to be associated with disease. And so to be able to predict it and get it right, they typically are large genes with small exons are ones that are associated with disease. But being large genes is utterly trivial. Large genes are more likely to cause disease because they're large. It's as simple as that. There's nothing subtle about that effect.
Thanks. And if anyone's got questions online, they can post them in the chat. I mean, you refer to the sort of disease causing synonymous variants. Presumably they're just phenotype influencing. It's just that some of the phenotypes that one can get money to study diseases.
To what extent ClinVar is skewed by that, I don't know, but it has to be. I'm fairly confident that our databases are very heavily skewed, not necessarily simply by the monetary effects, but there's an interesting population genetic effect, which is that disease-causing mutations, the diseases of disease-causing mutations typically sit a little bit above the expected frequency at mutation selection equilibrium. Yeah, so you predict what frequency should they sit at, and there's a very nice, elegant explanation for that, which is that the field is indeed skewed to the more common diseases. So if you overstudy the more common diseases, the diseases that you know about will sit above mutation selection equilibrium. And so the other really strong predictor of, as I said, of the disease causing, the gene associated with being disease causing is simply the size of the gene, size of the CDS, because a small CDS will only rarely cause disease. And so we simply will not be picking it up. And they end up in this bin called rare diseases.
Yeah, that's the explanation. So look, there's we've gone past time, but there's any more questions, but
I just quickly ask, I mean, coming back to your starting opposition between your Swiss watch and the synonymous watch. I mean, you're arguing really in favour of the former, right?
The synonymous sites, so yeah, my view is that when you're looking at protein coding genes, we are missing part of the Swiss watch. Because if you overlook the synonymous sites, you're missing part of the Swiss watch. However, en masse, you wouldn't want to derive the same conclusion. What we see throughout the rest of the genome, I would say, is much more Mickey Mouse watch-ish. But if you only study the genes, then you're sort of migrating towards a Swiss watch by definition anyway. So yes, when looking at genes, I'd say there are cogs that we didn't understand the mechanisms of before, but we now do. But when you're looking en masse and you ask, well, why do we have large introns? Why we've got so much intergene spacer? That is much better explained as it's simply rubbish that happens when populations of sizes are small. Selection is not a very effective process. And so our ability to get rid of a transposable element insert is really rubbish.
Right. So that's why we've got massive long introns.
And I think we've got this funny little problem that we've got massive introns and it degrades the splicing process, and you get another bit, and it degrades the splicing process, and you get another bit. And at some point, it's degraded the splicing process so much that the selection is actually now strong enough on the synonymous side to say, stop it, please. But each one cannot itself be stopped. So it's an interesting ratcheting process. Of decay, do something about it, decay, do something about it. And so we exist as a sort of mutational drift equilibrium in which most of what we do is rubbish. And most of those splice forms are rubbish. I mean, I don't know, one of the most striking things, I've always wanted to see this particular problem really properly quantitated. So a couple of years ago, I did this funny little test. If you take a random bit of 100 nucleosides of human DNA, and you take our computational predictions for how many transcription factor binding sites we think are in there. So I did this a couple of years ago, and the answer was, I just couldn't believe it, because the answer is that about, on the average, 60 to 70% of all of that sequence within 100 random base pairs is transcription factor binding site. And there's about 15 on the average transcription factor binding sites within any random 100 base pairs. That cannot possibly, what that says is your genome is transcription factor binding site. Yeah. But one of the, I thought the most striking results of the science papers, the zoonomia thing that came out, was it last week, is they estimated that we've got over 15 million transcription factor binding sites in the human genome, with evidence of sequence conservation for about 2 million of them. So that means that the vast majority of places that transcription factors are binding and making a transcript from are not being conserved. And when you then look at the transcripts, they have all the fingerprints are just utter rubbish. RNA pol 2 falls off, so you get a small fragment. There's something else coming off the other way. It's a small fragment. They go as far as an intron, and we've actually got a specialist machinery to see a transcript which isn't ending properly. It's ending in part of an intron. Just digest those. We've got an incredibly effective series of filters to go, you're crap, you're crap, you're crap, you're crap. And part of this nuclear export is actually because most of what is in the nucleus is utter rubbish. And our real challenge as humans is getting the rubies, those very rare, lovely transcripts that we actually want, getting those out to the ribosomes. Okay, you can now be processed, please. Even after they get out, we keep still making mistakes, and we've got P-bodies on our cytoplasm. And the P-bodies accumulate UA-rich transcripts. And UA-rich transcripts are just the sort you'd expect. So our mutation virus is GC2AT. So what rubbish is going to look like is it's going to be intronless and AT-rich. And P-bodies are sort of the last attempt to capture UA-rich stuff, which looks as though it's complete crap before it gets to the ribosome. And actually going back to the point about the tRNAs, we do actually have a surveillance system on our ribosome, such that if a transcript has been processed too slowly, we actually abort the whole process. And codon usage affects that as well. So again, this is, you're a lucky transcript. You weren't destroyed at source, you got out of the nucleus, you were put into P-body, you made it to the ribosome, you lucky bugger. But you're going too slowly, so I'm not allowing you to go anywhere. And of course, and then we've got NMD, which goes, your stop codon is not in the right place. You're out of here. We'll give you one round, and then you're out of here. So the whole thing is basically wired because we're rubbish.
There's one quick question online, which that'll be the last question, which is from Amy: do current tools for SNP deleteriousness prediction presently appropriately score the consequences of these synonymous mutations?
I don't know any other than ours that actually says, are you disrupting an ESE per se? There are some that have incorporated the machine learning splicing disruption.
Yeah.
And of course, if you ask, what does the machine learning splice disruption actually predict? It is actually, you can backtrack it and it's pulling out ESCs. And so it's known that those things are looking at mutations that have disrupted ESCs in no small part, but not only those, they also cover splice site mutations and things like that. But there's also there are some ESSs, those are exonic splice suppressors, but we find no evolution signals and nothing in the machine learning approaches to splicing seem to detect those either. So yeah, so not explicitly, but implicitly from the machine learning approaches that have been glued into some of them.
Great. Well, thank Laurence for a really interesting talk. Thank you.